So I'd first like to do the acknowledgement of country and would like to acknowledge and pay respects to the traditional owners of the land in which we meet, the people of the Eora Nation. It is upon their ancestral lands that the University of Sydney is built, and also wherever you are out there on Zoom. As we share our own knowledge, teaching, learning and research practices within this university, we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country.
I'll start by giving an overview of the ABCD dataset for anyone who's not familiar with it, and then I'll talk through the study that we've done looking at alcohol use during pregnancy and child outcomes. So a little bit about the study. So back in 2015, the National Institute of Health in the US funded the ABCD study, and it's the largest long-term study of brain development and child health in the US. So over 2 years, they recruited nearly 12,000 students and their families to be involved in the study from all over the US. And you can see here on the screen, there's 21 different research sites. So the participants reflect the US population of boys and girls and include students of diverse races and ethnicities, different education and income levels, and different living environments. In terms of recruitment, they identified all of the schools within a reasonable travel distance from each of the research sites and then selected a subset of schools for enrolment. The principals got sent information packs, which they then passed on to all of the families of 8, 9 and 10-year-old students. And then from there onwards, all contact was directly between the ABCD researchers and the families. So all of the data that's collected as part of this study is freely available for anyone in the research community to use. You just need to request access to the data from the NIH, and then you're able to download all of it from there. The data gets released once a year, usually around August. So the first release was in 2018, and they included about half of the participants' baseline data. And then the most recent release was August 2019, which included all of the baseline data and approximately half of the samples 6 and 12-month follow-up data. So how I became involved in this study was I visited 2 of the research sites last year for a few months, thanks to funding from the Matilda Center and Premise. So I first went to the leading research site at the University of California in San Diego. And then I also spent some time at the Medical University of South Carolina. And since then, myself and my supervisors, so Louise Newton, Marie and Maxine, we've been collaborating with ABCD researchers on some papers. So this is an overview of the study protocol that the kids and their caregivers do at each visit. So when the child first enrolls in the study, they complete a 6 to 7 hour research protocol. This includes surveys, they do a clinical interview, cognitive tasks on an iPad, they provide biosamples of their blood, they get some hair cut off their head, and they give saliva samples. They also do a 2-hour brain scan, and they can either do this in 1 session or over 2 sessions. So when I was at UCSD, I sat in on one of the sessions, and it is a seriously long procedure. The RAs do a really great job of keeping the kids interested and entertained with snacks and games. They take lots of breaks throughout the survey, but yeah, it's huge. And at the same time that the kids are doing all of the surveys, another RA will interview the caregiver, which is either the biological or adoptive parent. So they also complete surveys and clinical interviews, which take approximately 3 hours. So the brain imaging and biospecimen collection are done every 2nd year, and then all of the remaining assessments are conducted semi-annually or annually. So you can see the other protocols are slightly shorter. And at the moment, I believe they've got funding up until, so to keep doing this up until the kids are 20. But I believe that they're pushing to hopefully continue following them up for longer. So here's a quick overview of the type of information being collected from the youth, their caregiver, and from other sources. So there are assessments of neurocognition, physical and mental health, social and emotional functions, and culture and family environment. The kids also undergo multimodal structural and functional brain imaging and biospecimen collection. So I mentioned before that they do bloods, saliva and hair, and then also whenever the kids' baby teeth fall out, the parents will send them in as well. I think that's for genetic and epigenetic analyses. So overall, there's a huge amount of data available. When you get access from NIH, you download 230 separate files worth of data. And from memory, I think if you were to combine data from all of the files, you would end up with more than 80,000 data points per participant. And this is just for baseline and the 6 and 12-month follow-up. So it's really going to be huge over the next couple of years when there's more data available. And when you get access to this, it's definitely a bit daunting at first, trying to work out what it all means. But it's once you work out how to use the study dictionaries, it's not too bad finding the data that you're interested in exploring. So if anyone's interested in learning more about this or wanting to get access to the data, please let me know, and I can walk you through it to hopefully save you some time. So since 2017, there's been 49 ABCD publications, including 32 research papers, and 34% of these research papers have come from investigators that are not involved in the study. So it really is open for anyone to use. I've shown just a small selection of papers on the slide here. And as you can see, the research questions widely vary, showing the wealth of data that's available to explore. So there's been some studies on suicide, lead exposure, eating disorders, screen time, a huge range. So I'm going to move on now to the study that we've conducted. So this was a collaboration between the Matilda Centre, UCSD and MUSC. So here we examined alcohol use during pregnancy and the associated psychological, behavioural and neurodevelopmental outcomes in kids. So a little bit of background to this study. The global prevalence rate of alcohol use in pregnancy is approximately 10%. The dose and exposure patterns of alcohol use greatly varies between women, and recent estimates suggest, published in The Lancet and JAMA, suggest that approximately 1 of every 13 women who consumed alcohol during pregnancy delivered a child with fetal alcohol spectrum disorder. Now, FASD is an umbrella term used to describe individuals within the overarching category of prenatal alcohol exposure. So it includes fetal alcohol syndrome, which is the most severe outcome of drinking during pregnancy, but it also includes partial FAS, alcohol-related neurodevelopment disorder, fetal alcohol effects, and alcohol-related birth defects, and these range on a spectrum. So generally, children with FASD were exposed to quite heavy levels of alcohol use during pregnancy. And there's a lot of literature to suggest that FASD is associated with brain anomalies, postnatal growth restriction, distinctive facial features, as well as psychological, cognitive, and behavioural deficits from birth through adulthood. Now, for every 1 child diagnosed with FASD, there's approximately 12 alcohol-exposed children who don't meet criteria, which lends the question, what are the effects of lighter levels of alcohol use during pregnancy on child outcomes? And this is what we set out to explore using the ABCD data. So first, we wanted to know, do psychological, behavioural and neurodevelopmental outcomes differ in children aged 9 to 10 between those who were and weren't exposed to alcohol in utero? So we first used a dichotomous variable to look at this, and then we followed it up on significant effects to see whether greater doses across pregnancy were associated with poor outcomes. So to see whether there were dose-dependent associations. Next, we wanted to know what the common patterns of alcohol use were during pregnancy and whether those patterns were associated with adverse outcomes. And lastly, we wanted to know whether regions of the brain mediated associations between alcohol exposure and any psychological and behavioural outcomes that we found. So very quickly, just an overview of the methods. So we used baseline and 1-year follow-up data on psychological, behavioural and neurodevelopmental measures from the ABCD dataset. So we had cross-sectional baseline data for all of these outcomes, and then for approximately half of the sample, so for 4,169 participants, we had follow-up data on psychopathology from the Child Behaviour Checklist, and also on lifetime mental disorder diagnoses from the KSAD. So we ran generalized additive mixed models and also multi-level mediation models to explore the data. And we controlled for a number of birth-related, genetic, maternal, and socioeconomic status covariates. So there were 13 in total. And then in terms of random effects, we nested the individual within their family, as there are siblings, twins, and triplets in the study. And then we nested family within the research site. So in the ABCD cohort, 26% of the children were exposed to alcohol in utero. Interestingly, there are really similar birth weights and rates of premature births between each group. Maternal age is also very similar, and the exposed group's parents on average had a high level of parent education than the unexposed group. Now, I also want to highlight here the range in total drinks consumed. So it ranged from 0 up to 80 drinks once we converted outliers. So this isn't capturing the patterns of use, rather just the total amount. But just wanting to highlight that this is very light levels of alcohol use that we're looking at here. In terms of other in utero exposure, those with alcohol exposure were also significantly more likely to be exposed to tobacco, cannabis, and cocaine than youth with no alcohol exposure. And the results that I'm about to show you, we ran sensitivity analyses where we matched participants in each group, so those who weren't exposed and those who were exposed. We matched them on all of the demographic variables that we were including, and excluded all participants with any other prenatal substance use exposure, so that we could be more confident that the results that we were finding were related to alcohol rather than these other substances driving the effects. So when we looked at the data dichotomously, so splitting those with and without exposure, we found that youth with prenatal alcohol exposure exhibited significantly greater psychopathology. They were more likely to have a lifetime diagnosis of oppositional defiant disorder and separation anxiety. They were more impulsive, and they had attention deficits compared to youth who were not exposed to alcohol. We also found that a number of these associations were dose dependent. So greater levels of alcohol use during pregnancy were associated with greater problems in youth. In terms of the brain indices, we found that prenatal alcohol exposure was associated with greater global and regional cortical volume, and also with greater cortical surface area. And again, a number of these associations were dose dependent, where greater levels of alcohol use were associated with higher volume and higher surface area. So next, we wanted to take a deeper dive into the common types of alcohol use patterns during pregnancy and see whether all or just some of these patterns were associated with adverse outcomes, and to determine whether there was any level of alcohol use that could be deemed safe for the fetus in terms of later psychological, behavioural, and neurodevelopmental outcomes. So to group participants into different exposure patterns, we used an established prenatal alcohol use classification system by the team at Telethon Institute in WA. So as you can see on the slide, for example, a woman would be classified as a light drinker if she had 1 or 2 drinks per occasion and less than 7 drinks over the week, while a woman who had 1 or 2 per occasion but more than 7 over the week would be classified as a heavy drinker, and so on. So in the ABCD study, the caregiver, which nearly 90% of the time was the biological mother, she was asked about alcohol use before and after she found out she was pregnant. And so if the caregiver who was interviewed was the biological father or an adoptive parent, they were also asked about the biological mum's alcohol use patterns, and if they weren't sure, those youth weren't included in the analyses. So we applied the alcohol use classification system from the previous slide to patterns of use before and then after knowing of pregnancy. And then based on this information, we created 6 groups. So women were classified as abstinent if they didn't report any alcohol use before or after knowing that they were pregnant. They were classified as a light reducer if they reported light levels of use before they knew they were pregnant and then were abstinent once they knew. They were classified as light stable drinkers if they reported light use throughout the whole of pregnancy. And then for heavier reducers, because of numbers, we grouped all women who reported either moderate, heavy, or binge levels before knowing, and then either light or no use afterwards as heavier reducers. Whereas if they kept this similar pattern throughout, they were heavier stable drinkers. And then finally, if they reported more alcohol use after knowing that they were pregnant compared to beforehand, they were classified as an increaser. So overall, 76% were abstinent, 14% were light reducers, 1% were light stable drinkers, 9% were heavier reducers, and 0.2 and 0.3% were either heavier stable or increases. So because those bottom 2 categories had such low numbers, they weren't included in the analyses, and we just stuck with the 4 top group for the exposure pattern analyses. And that accounted for 98.1% of the sample. So on average, light reducer mothers consumed 2.3 drinks per week for the first 7 weeks of pregnancy, and that the 7-week mark was the average time that women found out they were pregnant. And this equated to approximately 16 total drinks across their whole pregnancy. Stable light drinking mothers consumed approximately 1.1 drinks per week throughout their entire pregnancy, and this equated to approximately 44 total drinks. While heavier reducer mothers consumed approximately 5.3 drinks per week for the first 7 weeks of pregnancy, and then the vast majority of the samples stopped drinking once they knew that they were pregnant. So on average, they consumed around 36 drinks throughout pregnancy. Now, for these heavier reducers, we did split the analyses into mothers who became abstinent once they knew they were pregnant versus remained light drinkers. And results were consistent between those 2 groups, but the number of women who became light drinkers was really low. I think it was around 30 or 40 participants. So because the results were consistent, we kept those mothers in the same group. So compared to unexposed kids, all of the exposure groups exhibited greater psychological and behavioural problems, varying mental disorders. So again, we saw separation anxiety and oppositional defiant disorder. And then the heavier reducers, we saw a greater likelihood of the youth being diagnosed with attention deficit hyperactivity disorder. And then to complement the dose response analyses, when we compared the light and heavier reducers, we found that children of heavier reducers also reported greater withdrawn and depressed behaviour, attention deficits, rule-breaking behaviour, and aggression compared to youth of light reducers. And again, we found that all groups reported differences in volume and surface area compared to the unexposed youth. So the takeaway here is that all exposure patterns, including the lightest levels of use, were all associated with poorer child outcomes at age 10. So the final thing that we wanted to look at was whether the brain differences that we observed partly mediated the associations between prenatal alcohol exposure and the adverse psychological and behavioural outcomes. And we found that this was the case. So varying regions of the brain partly mediated all of the associations at baseline, where we had data on all of the measures, and then also the psychological and mental disorder associations, where we had the 1-year follow-up data. So here are some of the limitations of the study. There was potential maternal underreporting of alcohol use during pregnancy. The parents were when they were interviewed, were being asked about alcohol use patterns from 10 years ago. So it could be imprecise. The ABC dataset also does not provide data on trimester-specific alcohol exposure. However, because it is retrospective, the quality and the confidence we would have in those analyses might not be there anyway. There were a few cases of youth who were exposed to stable light or heavy drinking or increased consumption throughout pregnancy, although our interests were in light levels anyway, and there is a lot more literature around heavier levels of drinking. And then finally, we didn't assess the impact of preconception paternal alcohol exposure on child outcomes. And over the last few years, there has been some preliminary evidence to suggest that there could be an association there. So our overall conclusions from this study were that relatively light levels of prenatal alcohol exposure was significantly associated with greater psychological and behavioural problems in youth. Exposure was related to greater global and regional brain volume and greater regional surface area. And these brain differences partly mediated the above associations. And finally, examination of dose-dependent relationships and light alcohol exposure patterns during pregnancy suggests that women should continue to be advised to abstain from alcohol consumption during pregnancy. So this was the study that I wanted to go through in detail. It actually got accepted for publication this morning, which was exciting. And it's the first study to look at really light levels of alcohol use in a large sample. So I think the findings will be important for families, clinicians, and policymakers moving forward. I also just wanted to give an overview of some of the other papers that have either been published or are under review or in preparation, that are collaborations between the Matilda Centre and ABCD researchers. So one area that we're looking at is parental history of alcohol and substance use problems. So there's a growing literature that's interested in determining whether there are brain indicators or I guess vulnerability markers that can indicate youth who are more likely to start drinking at a young age and be problematic drinkers. So we did a study looking at youth whose parents have alcohol problems or alcohol use problems, and we found that those youth had altered activation in inhibitory networks. Now, this was just using the baseline data and all youth were substance naive in the analyses. So this is going to be an ongoing project that's a collaboration with UCSD, where as some of the youth start drinking, we'll have a better idea whether these altered activation patterns in these inhibitory networks are good predictors of alcohol uptake in adolescents. And so another thing that we're looking at is predicting early alcohol use in youth. So we just ran the analyses last week for this and found that youth where their parents have a substance use problem and they also have high levels of family conflict in their home were more likely to have started drinking alcohol by the age of 10. And also for youth where their parents have problems with substance use and the youth has a lifetime diagnosis of conduct disorder. This was a similar case. They were more likely to have tried alcohol at baseline and at the 6 and 1 year follow-up. So another area that we are working on is looking at neural correlates of general psychopathology. So Louise Mewton is leading a study at the moment looking at psychopathology and brain structure, and she has found that general psychopathology and the dimensions of psychopathology, so internalizing, externalizing, and thought dimensions, were all linked to lower global volume and surface area in pre-adolescence. And we're now just running a follow-up study to that one, looking at functional networks. And we've found, again, that general psychopathology and the 3 dimensions are linked to altered connectivity between the executive control networks within the brain. And then the last one that I wanted to point out was a study doing looking at screen media time. This is a bit different to the other ones. And we've found that screen media time is largely unrelated to involvement in co-curricular activities, so sports, crafts, musical instruments at the age of 10. And that is a collaboration with the Laureate Institute in the US. So I guess overall, just wanting to highlight that there are a huge number of measures included in the data set that's available, and you really have unlimited options in what you can explore. So I just wanted to say a huge thank you to my PhD supervisors, Louise, Marie, and Lexine. And these are all of the ABCD collaborators that we've been working with, and then also other collaborators. So we've had Matt and Tim involved, Andrew, Miri, Bob, and Forrest at UNSW. So thanks.
Great, thanks very much. That was an awesome presentation. It's amazing how much data that single study has and is going to collect. And it really is a valuable resource that I'm glad that people like you and Lou Mewton are making use of it. So I'd just like to open it up to everyone now to ask questions using the Q&A function of Zoom. And while people are typing in questions, I've got one more about the kind of the logistics behind getting access to the data, and whether or not there is kind of like a single organization that tracks what everyone is doing with the data. In order to get access to the data, do you have to submit a project plan? I'm sure you do. And then do they keep track of who's doing what, so you can go to that resource and then check to see if you have an idea whether that's already been done or yeah.
It's a little bit disjointed. So you need to submit sort of a research protocol to the NIMH, and I'm not even sure if they read it. You pretty much get it accepted straight away. They are encouraging researchers to pre-register their paper on Open Science Framework, but again, that seems really inconsistent. On the ABCD website, they are listing all publications that have come out, so you can keep track there, and that seems to be quite up to date. But so far, there doesn't seem to be a consistent way of seeing what people are actually working on at the moment. Within the ABCD investigators, they have an internal system where they can track what each other are doing, but people who aren't involved or who aren't a chief investigator on the study can't see that. So yeah, a little bit of a mess.
Cool, so you just kind of have to hope that no one else is working on the same thing as you are at the same time, or at least you need to work faster and get it out in a hurry.
There's a huge push. So we were working with ABCD collaborators, and there's a huge push to work as fast as you can because of that reason, to just try and get things published before others, because there's no consistent way of seeing what everyone's doing.
Yeah, I guess it's good motivation.
Yeah.
So a question from Paul. He says, great presentation, larger brain volume and surface area sounds good, but apparently it's not.
Yeah, this was a tricky part of that study. So yes, normally larger brain volume and surface area is a good thing. And normally when you would look at studies with adults where you see, like if you looked at drinking in adults, you would see that they have smaller brain volume and less surface area. But what we think is going on here is throughout adolescence, and especially early adolescence, that is when brain size peaks, and then you start seeing a drop off throughout adolescence. So what we think could be happening here, and we need to look at this longitudinally, is that possibly these kids have got delayed development. So they're kids who weren't exposed, their normal developmental trajectories are happening where their brains are starting to decrease in size, and they've got healthy pruning going on and myelination, whereas for these kids, potentially that has been delayed, possibly. And also, the other part that was tricky here is in FASD kids who are exposed to much heavier levels than what we saw here, they also report lower brain volume and surface area. So for some regions of the brain, we did start seeing an inverted U shape. So for the much heavier levels of alcohol use, we started seeing drops in volume and surface area. So that's definitely a tricky part. And I think we need longitudinal data to better decipher what's happening. But I guess the critical point of the paper was that the larger brain volume and surface area was linked to poorer psychological and behavioural outcomes in kids exposed to alcohol.
Okay, cool. Emma's got a question. She says, awesome presentation and really novel and important findings for light use during pregnancy. Is there any indication whether the higher levels of psychopathology, behavioural issues, etc., are at clinically relevant levels?
Sorry, I'm just reading that question again. Is it, high levels are at clinical? Yeah, so they were definitely subtle effects, and at higher levels, we did start seeing some that were at clinically relevant levels at the higher end of the spectrum, and also with seeing those KSAD mental disorder diagnoses, higher prevalence. But yes, overall, they were definitely subtle yet significant differences between the groups. And you're seeing those dose-dependent associations between levels of alcohol use and the severity of those psychopathology and behavioural outcomes.
Cool. Well, they're really not findings. And you mentioned that those results that you presented were published?
Yes.
Where are they published? Because you left that out.
In the, well, not published, it's been accepted.
Accepted, yep.
Into the American Journal of Psychiatry.
Yeah, which in itself is amazing, and you need to be congratulated for that, because that's one of the top psychiatric journals in the world, and it's really hard to get into with a massive impact factor. So well done for that.
Thank you. Yeah, it was seriously tough.
Yeah, I can imagine.
We submitted in November, and it went through 3 iterations or 4 iterations of review. The first round, it was 9 pages of reviewer comments from 5 different reviewers.
Yeah, so that basically you have to rewrite the whole paper in response to those reviewers.
Yes. Yeah. So when I first submitted, I was only looking at the brain indices, not at psychopathology or behavioural outcomes. And something that is probably good to know with the ABCD data is because reviewers know how much data that is available, you will just get asked to include everything. That's happened multiple times now. They just want more and more and more, which, yeah, it's definitely a better paper. So in this case, it's worked out well. Yeah,
definitely. One more question from Paul. He says, why no biosamples from the caregivers when the kids are so heavily sampled?
Biosamples is in the hair. Yeah, I'm not sure. They're definitely more interested in, I guess the aims of the study are looking at child development. So the questions that parents asked about, they are asked about family history related to a whole range of things, alcohol use, psychopathology, family environment, but everything is, I guess, directed at questions around the youth, and that is their focus of the study.
Cool. Okay. Yeah, he just clarified, yeah, hair and blood for genetics.
Yeah, I'm pretty sure that the parents don't get asked to provide that, but I'm not 100% sure.
Very cool. All right, one from Erin as well. Are there any similar neurodevelopmental studies that look at alcohol and immune function together?
I'm not sure off the top of my head. So I'm sorry, I would have to have a look into that.
Okay. And one from Marie. What are the recent findings on paternal influence of these outcomes?
Okay, so I looked at this quite a while ago, but I think it was, I think it's just been one or maybe a couple of studies that have found alcohol use in the father in the preconceptual period of, I think, a couple of months was related to some outcomes in babies and young children. I don't have any specific details on that, I'm afraid. I can't remember.
Okay, no worries. All right, there doesn't seem to be that there's any other questions. So thank you very much. Oh, wait, one from Scarlett, last one. Such an interesting presentation, concerning that even children of women who stopped drinking once they found out that they were pregnant had worse outcomes. Will part of your work include any recommendations related to this?
Yeah, I think this is tricky, and it's a really, I don't know if touch is the right word. Yes, difficult to, I guess, discuss and recommend it. I guess it's more helpful with developing recommendations for people who are planning pregnancies, and they're able to take these findings on board, but much more difficult for people with unplanned pregnancies. And I guess how to address that, yeah, that's something that we've been talking about and need to keep talking about, because that is definitely a difficult part of these findings.
Cool. One from Lexine. She says, great presentation, Brianna. I know we can't be sure due to the study design, but from the timing we think that the drinking probably happened in trimester 1, right? In which case is the message to encourage women to stop drinking prior to trying to get pregnant?
Yeah, exactly. So on that same point, I think that will be important messaging for people who are planning to conceive and have a child, and then more difficult for unplanned pregnancies. But yes, that's correct.
Oh, very cool. All right. There's no more questions that come in unless some last minute ones come through, but thank you very much. Really great work, and amazing to hear that it's been published in American Journal of Psychiatry. You should go out and have some drink. Well, you can't really go out anywhere, but you should celebrate at least a little bit with those people in your household for successfully getting through that review process and published there.